Today, I am taking up the topic transcription. Transcription is the first phase of gene action or gene expression. You may ask, what is a transcription? For example, when you take up a original document, we call it as a script. If you transfer the information present on the original document onto a copy, or when you make a copy of the original document, it is known as transcript, and the process is known as transcription. So the same term is applied here. Transcription means copying the information from DNA strand to mRNA strand. That is about transcription. It is a very complicated process. So, transcription means, in genetics, copying of information from one strand of DNA onto the mRNA strand or messenger RNA. Then, this transcription process is governed by the principle of complementarity. You know that in DNA, the two strands have complementary nitrogen bases. That rule applies here. But the only difference between replication of DNA and transcription is, in DNA, the same complementary nitrogen bases are present. For example, adenine combines with thymine, guanine combines with cytosine. But coming to transcription, in the case of transcription, in the place of thymine, uracil is incorporated. That is the only difference. Then, how this information is copied from DNA to mRNA? You know that DNA is a double helical structure having two strands running anti-parallel to each other. It is, or traditionally, when you draw a DNA, the DNA strand with 5-3 strand is written like that on the above, and 3-5 polarity strand is written below that one. So, traditionally, when you draw a DNA structure, the strand with 5-3 polarity is written on the top, and the strand with 3-5 polarity is written below that one. So let us say these are the two strands of DNA molecule that has to transcribe. Now, this strand drawn in red color has a polarity in the direction of 5-3. The strand drawn in black ink has a polarity of 3-5. There are some rules about transcription. The mRNA strand is always synthesized on the strand with 3-5 polarity. So mRNA strand is always synthesized on a strand with 3-5 polarity. And the synthesis of mRNA molecule, mRNA means messenger RNA molecule, always takes place in 5-3 direction. This is very important. It is just like replication of DNA. In the case of DNA also, the replication process proceeds from 5-3 direction. Here also, synthesis of mRNA takes place in 5-3 direction. Then, the question is, which strand involves in transcription? It depends upon the message the mRNA has to copy. I have told you, transcription always occurs on the strand with 3-5 polarity. Let us say this is the strand on which transcription is about to take place. If you draw the base pairs of this segment of DNA, let us say it looks like this. This is a strand with 5-3 polarity. The bases are T, A, C, G, C, G, G, TA, CG, G, TC, A. Their complementary strands are A, that means thymine combines with adenine, adenine combines with thymine, cytosine combines with guanine, like that. Here we find complementary nitrogen bases. The purpose of transcription is to copy the information present on 5'3" strand, this strand. But if a copy is made from this strand, we get this message. But the message we want is this one. To get this message, we have to make a transcript from this strand. See, if you make a transcript from this strand, what you get? U, A, C, G, C, like that. I have told you, in the case of RNA, thymine is replaced by uracil. So here, instead of thymine, uracil is present. So T, U, A, C, C, G, G, C, C, like that. Again here T, you find U. So this is similar to this strand. So in the case of transcription, if you want to make a copy of this sequence, we have to copy it from this one, or we have to transcribe from this strand. Then only we get this copy. That's why we have to remember some terms here. The strand from which mRNA is synthesized is known as template strand. So this is the template strand. The strand in 3'5" direction is known as template strand. The strand in 5'3" direction is known as non-template strand. This one. So this is the template strand. This is non-template strand. Then there are also two alternative terms. The template strand is also known as antisense or non-coding strand, whereas this strand is known as non-template, sense strand or coding strand. Why this is called sense strand? Why this is called a non-sense strand? The aim of the transcription is to copy this information. So it must make this sense. The sense present on the strand with the 5'3" polarity. So this strand is known as sense strand or coding strand. And this is known as antisense strand or non-coding strand, because the coded information is this one, not this one. Though it is a copy is made from this strand, the coded information is from this strand. So this is known as coding strand or sense strand or non-template strand. This strand in 3'5" direction from which mRNA made is known as template strand or antisense strand or non-coding strand. I think these terms are very clear to you. Don't get confused. Then, so in this case, this is the mRNA synthesized from this strand, 3'5" direction strand. So here this is the mRNA. We call it as mRNA transcript from the DNA strand. Then, suppose if both the strands are involved in transcription, what happens? There is a danger, because two complementary mRNA strands are made. And because of the complementarity, both the mRNA strands join and a double helix of mRNA strand is formed. So, to avoid this danger, only one mRNA strand is produced from the template strand. That is very important. Then, the transcriptional unit of DNA consists of 3 parts. This is the transcriptional unit. It has a structural gene, this one, this segment, and also a promoter ahead of the sexual gene and a terminator after the sexual gene. So, the unit of transcription has three parts. This is the promoter, this is the structural gene, this is the terminator. The promoter is always, it is towards the right of the sexual gene, towards the left of the sexual gene. Terminator is always towards the right of the sexual gene. Then, the nitrogen bases or the base sequence, nitrogen base sequence present ahead of the promoter are known as upstream sequences. Similarly, the base sequences present after terminator are known as after the promoter are known as downstream sequences. So, these are the upstream sequences, these are the downstream sequences. Then this is the strand 5-3 polarity, and this is the one with 3-5 polarity. Always transcription takes place on this strand with 3-5 polarity. Then we use two terms, cistron and gene, to convey the same meaning. Though we use them interchangeably, they are quite different. A gene, genes are units of heredity. They participate in transcription and they also participate in replication of DNA. Whereas a cistron is concerned with the synthesis of a polypeptide molecule, the synthesis of a polypeptide molecule. So, they are different functionally. Then, the genes participating in protein synthesis or polypeptide synthesis are known as structural genes. So, mRNA is made from the structural genes. Then, transcription of mRNA is different in prokaryotes and eukaryotes. You know that in prokaryotes there is no nuclear membrane. So transcription and translation goes on simultaneously in prokaryotes. In eukaryotes it is not the case. Chromosomes are confined to the nucleus. So transcription takes place inside the nucleus and translation takes place in the cytoplasm. So that is the main difference between prokaryotes and eukaryotes. Then the enzymes involved in transcription are also different in prokaryotes and eukaryotes. In prokaryotes, there is one enzyme known as DNA-dependent RNA polymerase, or simply RNA polymerase. This enzyme involves in all the three processes of transcription, that is, initiation, elongation, and termination of mRNA molecule. For initiation of transcription in prokaryotes, the mRNA is associated with a sigma factor. So for initiation, DNA-dependent mRNA polymerase transiently associates, transiently means temporarily, associates with sigma factor. For termination, it transiently associates with another factor, which is known as rho factor. So sigma factor along with RNA polymerase helps in initiation or in the identification of the promoter, and RNA polymerase along with rho factor participates in termination or identification of the terminator. That we should not forget. Then coming to eukaryotes. In eukaryotes, 3 types of RNAs are present. They are RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. RNA polymerase 1 is responsible for transcribing mRNAs for synthesis of, or we can say in other words, it transcribes rRNAs. It transcribes rRNAs. R means ribosomal RNA or precursors of mRNA. Then, so it transcribes for ribosomal RNAs and also 28S, 18S and 5.8S proteins in the nucleolus. So, the action of RNA polymerase 1 is in the nucleolus. In the nucleolus, it participates in the transcription of ribosomal RNAs, 28S, 18S and 5.8S. Then RNA polymerase 2, it transcribes HNRNA or precursor of mRNA in the nucleoplasm, not in the nucleolus, but in the nucleoplasm. HNRNA means heterogeneous nuclear RNA. Heterogeneous nuclear RNA. This is the heterogeneous nuclear RNA. Heterogeneous nuclear RNA has 2 components, introns and exons. Introns serve no purpose. Only exons participate in translation. So, the heterogeneous nuclear RNA or mRNA has to be processed. This process involves removal of these introns. When you remove these introns from the heterogeneous nuclear RNA, mRNA, we get this. This is processed mRNA. This processed mRNA is released into the cytoplasm. So, removal of introns is known as splicing. The process of removal of introns is known as splicing. Splicing occurs in the nucleus. Then, the third enzyme is RNA polymerase 3. It transcribes transfer RNAs, srRNAs and snRNAs. srRNA means small regulatory RNAs. snRNAs means small nuclear RNAs. So in this way, these 3 polymerases have 3 different functions. So again I repeat, RNA polymerase 1 transcribes ribosomal RNAs, that is 28S, 18S and 5.S in the nucleolus. Whereas RNA polymerase 2 transcribes heterogeneous nuclear RNA or precursor of mRNA, which has both introns and exons, and the exons are retained, introns are spliced out in the nucleus. After processing, the remaining DNA is known as processed mRNA. Processed RNA is known as processed mRNA. Coming to RNA polymerase, its action is also in the nucleoplasm. It transcribes tRNAs, sRNAs and snRNAs. Then, after the processing of mRNA, there is one more step remaining to make it functional. To make the mRNA functional, a cap has to be added at the beginning and a tail has to be added at the end. Cap is added at the 5 end, tail is added at the 3 end. Cap is added in the form of guanosine triphosphate. Guanosine triphosphate is a unique nucleotide. At the end at the tail consists of poly-U. A number of uracil bases are added. So it's called poly-U. In this way, the processed mRNA is converted into functional mRNA by adding a cap at the 5 end and by adding a tail at the 3 end. That is about the transcription process in eukaryotes and prokaryotes. Thank you.